Thanks a lot, Anna Rita. So I'm really excited to have both of these speakers come and present to folks here at AgBioData. I actually heard these talks at a recent Gene Ontology Consortium meeting, and I thought that the information that was presented by both Paul and Alex was really interesting and very relevant to the community at AgBioData. So first we're going to hear from Paul, and then we're going to hear from Alex. So our first speaker is Paul Thomas from the University of Southern California. He's a PI for the GO Consortium and also for the Alliance for Genome Resources, right? Anyway, he has a wonderful presentation that he's going to share with you. And please be a little understanding because I know he's having some connection issues. So hopefully it'll all work out.
Okay, yeah, amazingly, the timing turn. It looks like my power might be back on now. So yeah, this has been California for a little while. Okay, so thank you so much, Leonore, and thanks for the invitation. I'm really happy to talk to you about this. This really represents a lot of work by a lot of people over a long period of time, which of course I'm going to make an attempt to compress into like 15 minutes ish. So let's see if I can do that. Okay. So just a little bit of background, right? So the idea is that what we want to be able to provide is Go annotations for any genome. Okay, as probably most people on this call are aware, the gene ontology annotations are the standard functional annotations in the community. Here's a quick example of some of the main information that you get out of an example gene ontology annotation. It basically specifies a gene and then a functional characteristic, you could say, of the product of that gene, in this case, the protein product of the frizzled 1 gene in humans. And then it's associated with a type of evidence. There's always a type of evidence for every gene ontology annotation, every assertion, and a link to the reference that describes that evidence. So to date, 12 genomes have been, I'd say, highly annotated with Go function terms from experimental evidence taken from the scientific literature. So, these are all the model organisms that everybody is familiar with, including Arabidopsis. And so what do you do if you want to get gene ontology annotations for other sequenced genomes? So that's really what this talk is going to be about. Okay, so InterProScan has really been the most highly used tool for Go annotations for many years. It's been a source of these electronic, in other words, computational predictions of gene function Go annotations for many years, dating back to this very nice paper from Sarah Burge et al. Describing the process of basically how it works, that they assign Go terms to signatures in the InterPro database. So the InterPro database is a consortium of contributing databases, and I've shown all the members here. And each of them produces a signature, usually a hidden Markov model that can be used to identify hits to those HMMs. And if you've associated Go terms with these signatures, you can then assign those same Go terms to any sequence that hits those models. And so that's their basic idea, just to sort of go through their workflow, right? What's done by each of the member databases is that they will define a group of homologous proteins. They'll do that either by global homology, you would call that a protein family, or local homology, right, over part of the sequence. These are conserved protein domains. And in the case of protein families, these are most often very diverse families, but some databases like PANTHER have closely related subgroups in them, and those can sometimes give you more granular annotations, and I'll go into that in a second. But the basic idea then is to define, once you define a group of homologous proteins, you build an HMM or other signature using the sequences of the group members, And then at the InterPro Consortium Central Group, they have a curation group then that can assign to that HMM Go terms that at least seem to be in common to those characterized group members. As you know, most protein sequences have not been characterized, so they have to basically find the annotated group members, often in resources like Swissprot, and use that to assign Go terms. So then the second step, as I said, is once you have these annotated HMMs, you can score each sequence in any set of sequences, in any unannotated, any genome or set of sequences that doesn't have any Go terms associated with it. You can score each sequence then against the HMMs. You can collect all the HMM hits in all the different member databases for each of those query sequences, and then assign to each of the query sequences the Go terms that were associated with the HMM hits. Sorry for my misspelling there. And this has all been implemented in a very nice downloadable software package called InterProScan. So there are some limitations to this approach. Okay, so it's very accurate. It's been shown to be very accurate, but many of the Go terms are quite non-specific. The inherent limitation, of course, to the approach is that the Go terms, to be accurate, you have to make sure that the Go terms really do apply to the best of your knowledge of all of the sequences in a family. And so many protein families are large and diverse and can have diverse functions. And in that case, you can often just make a very, very general statement about the function. In some cases, you might just be able to say, it's an enzyme, right? It has catalytic activity, but I can't say exactly what that activity is. Okay, and so that's really sort of been the state of the art. So what I'm here to talk about, and what Alex will be giving you much more information about, is this new method that that integrates work by multiple groups to assign more precise and selective Go terms to sequences using InterProScan. And it integrates the work of the PANTHER group, which is also my project from my group. And this is basically a project to build phylogenetic trees, which really haven't previously been used in InterProScan. They've been using subfamilies derived from the trees, but not the trees themselves. The Go Consortium, and so there's been a long project starting in 2011 when we first sort of mapped out how this approach would work. To today, we finally have a large number of these models, but basically doing Go annotations, building evolutionary models of within trees. And I'll talk a little bit about that, but I don't have a lot of time. I'll try to give just a little more detail, but I can answer questions about it. But you can read a paper basically if you want details. And then InterPro, the InterPro Consortium main group, has implemented TreeGrafter and InterProScan. And so all taken together, we can now use annotated trees that can have a great deal of detail and can represent functions that are only present in some of the sequences in a protein family. Can represent this accurately, and then identify when a new sequence should really belong with a particular subgroup within a tree. And I'll give you some pictures that show this better. This later. Okay, and these are already available in the InterProScan output files for GoTerm, and they're labeled as PANTHER. Okay, so the basic approach is this, right? And this is what the Go Consortium has been doing for years, right? Which is to go from published literature to experimentally supported gene annotations, Go annotations. Okay, and here's what the PANTHER group has been doing for years, right? Which is to go from a group of reference proteomes, which are sampled based on the availability of function information, but also phylogenetic sampling, which tends to favor animals and plants, actually. And then build families for each of these. Each one has multiple sequence alignment, hidden Markov models, but most importantly for this project, phylogenetic trees. And there are 15,000 family trees across the tree of life now. And then combine those two. So this is the phylogenetic annotation project that I just talked about. in the Gene Ontology project, where the basic idea is to overlay experimentally supported Go annotations on a phylogenetic tree and use that to when functions first evolved in evolution and be able to localize that to a specific branch in a phylogenetic tree. So you can make branch-specific annotations of gains and losses of function that you basically are building a model of evolution in the family that then can be used. Once you have this model of evolution, you then can use the model whenever you have a gain, right? You have inheritance of that function through the tree, unless there's a loss. So we also model evolutionary loss of functions. And then, that Go term will be removed from a clade. Okay, and so here's sort of an example of how that works. So that's as much detail as I'll go into here. But again, you can read the paper to get more information, or I can answer questions. But at the end of the day, you end up with, we now have, for those 15,000 families, almost 10,000 of them have evolutionary models of selected Go terms through those families. Huge data set, okay? And so that's how we get sort of the basis. So the basis of this annotation system is all curated, reviewed annotations that take advantage of the massive corpus of annotations in Go, okay? All right, and then what do we do with it? All right, so the second approach, so the second part of the approach, of course, is I have to be able to annotate any arbitrary sequence. So now I've built something that I can use to annotate those 140 odd sequences or genomes that are in the trees. But what if your genome isn't in those trees? Okay, what can we do? Well, we can do it in InterProScan. We basically can graft a new sequence, and I'll show kind of how that works. Okay, well, first I'll give an example family, just why this is important. Okay, but yeah, I'll show sort of how the tree grafting works in a second. Okay, but basically this is the solution to annotating any arbitrary sequence. Okay, so an example of this is so just so you can get an idea of why this is important. Okay, here's a great example that we've mapped out the evolutionary model for. Okay, this is ubiquitin-like activating enzyme families. So ubiquitin-like molecules comprise things like ubiquitin, sumo, ATG8. You can sort of see the family. And when you reconstruct the evolution of this family, it's a really interesting story. Okay, that ancestrally, this family appears to have been just sulfotransferases. Okay, and that function, and so the model is based on what are our observations today. Okay, well, we have Go annotations saying that we've got a lot of bacterial sequences that are sulfotransferases. That's all we find among the bacterial members of this family. The archaea, not surprisingly, are completely unannotated functionally. Okay, but we have annotations in the eukaryotes, and there's one clade of eukaryotes actually that's still a sulfotransferase. Okay, so the most likely evolutionary model then is that the ancient function was sulfotransferase, which was inherited through all these billions of years, right, to modern day bacteria and modern day eukaryotes. Okay, but there was functional diversification in the family where sulfotransferase activity, according to our model, was lost, but you can see it was really modified, right? Because there's a sulfur group, in the cysteine of ubiquitin that allows it to be transferred. So you basically have this modification of an ancient sulfotransferase function that becomes a ubiquitin-like modifier function, and then subtypes of different ubiquitin-like modifiers, and then sometimes within a clade you can have later events distinguishing between yet more ubiquitin-like modifiers and different specificities of modern enzymes. Okay, so that's sort of what our model looks like. And so we can then, and so if I were trying to make a function annotation for an entire family in this case, I can really only say it's an enzyme. And in fact, that's what InterPro to Go would say about this. It does say about the PFAM family. Okay, so this will just give you an idea of there are many cases where this is a very valuable thing and you can get much more detailed annotations out of it. Okay, so how does TreeGrafter work? Okay, the basic idea is I've got this annotated tree. So think of it as branches in the evolutionary tree ending at a particular node have been annotated with Go functions. Each of these Go functions has to have experimental evidence somewhere in the tree among the descendants. So everything's traceable to evidence. Okay, but I have this model now. I have a phylogenetic tree, a model of how functions have been gained and lost. And now what I do is I computationally try to graft onto this tree, right, a new sequence. Okay, so let's say I've grafted at this point along this branch. Okay, what can I infer about the evolution of this? Or sorry, about what can I infer? So let's say I've grafted onto this branch here. What can I infer about? All right, okay, my animation totally didn't work when I transferred it. Sorry, guys. I'll just do all the animation here. All right, so the idea is, oh shucks. All right. Apologies. Okay, so I've grafted a sequence here. Okay, so what do I do then? I infer from the annotated node here which functions would be inherited to this branch of the tree. Okay, and this is what I can say about this sequence. Okay, so it gets these specific annotations from this part of the tree. Okay, but if I graft to a different place in the tree and I graft up here instead, okay, I'm going to inherit annotations obviously from this distant ancestor, but also a more recent ancestor, and I'm going to get a different set of annotations based on this graft point. Okay, so basically the idea is I graft new sequences computationally onto the tree, figure out from my evolutionary model what functions they would inherit based on their position in the tree. Okay, so it's a pretty nice system, and thanks to a lot of work by the especially Rob Finn and Matthias Blum at InterPro, it's quite efficient. So it's computationally efficient as well. So this is, I think, my last summary slide. Hopefully, I'm giving you enough time, Alex. And so how would you get these Go annotations from InterProScan? So again, InterProScan now includes both the InterPro to Go annotations, as well as the new annotations available from grafting onto the trees and taking advantage of these carefully annotated phylogenetic trees and these evolutionary models. You can either download InterProScan to run on any custom set of sequences. So this is available. You just got to make sure you have a recent version of InterProScan, and you turn on the -go terms option. But pre-computed results actually are now available for NCBI RefSeq. So NCBI has run this pipeline on all of the eukaryotic RefSeq genomes, except for the model organisms, which I advise you get the same way as you always have gotten from Go Central or the model organism database itself. And then Alex will talk about pre-computed results at Uniprode and sort of a little more detail about how these annotations compare to the annotations you're probably already familiar with out of InterProScan. So just have to thank, as I said, this has been a really big effort over many years from a lot of people, number of different funding sources. And so I just really want to call out, especially I just have to say, obviously I've put in all the different teams who've worked really hard on this. And this goes all the way from, the primary annotations where, without the substrate of the primary annotations, there would be no way to build these evolutionary models. The PanGo team that actually built the evolutionary models or built software that allowed these evolutionary models to be built and tracked. One thing I will say is that, if you find anything wrong with any of the annotations that come out of it, we've got a whole system for submitting and taking feedback on them and making rapid updates to the evolutionary models that then propagate through the whole system. So this is really designed to be a system that we continue to modify and improve over the years. And then, of course, I have to, and I think I've already mentioned the work of Rob and Matthias, but also Haiming Tang from my lab, who really did the original development of TreeGrafter and proved that this approach could actually work. And of course, as I said, groups all the way from InterPro, NCBI, and Unipro. So thanks for your time, and I can take a couple of questions now, or we can wait until after Alex has spoken.
Thank you. That's great. Do people have questions now, or should we just move on to Alex and then field questions after? Trying to see if I can see the screen. I don't see any hands up. So why don't we go ahead and Alex, why don't you give your presentation and then we'll do Q&A afterwards.
Sure. Thanks, Linda. Let me just share screen.
So, Alex is, just to give the introduction, that Alex is a senior database developer and part of the protein function development team at EMBL EBI, and he will give the next talk.
All right, everybody can see the screen okay?
Yep.
Okay, so basically, let me just introduce what Go is at EMBL EBI, because maybe some people is not familiar with this project, but what we basically do, we doing Go annotations of two different kinds. One is manual Go annotations, that what Paul described as the source of truth is something that people do for the very short, very limited number of species to kind of understand and the function or what processes they involve, where they're located, and our project is kind of a big collection of all these manual annotations. We have about 74 different sources for them, and we right now accounted for about 5 million annotations. The project is quite old. We started in 2001, and right now Protein to Go, our primary curation tool, has 1 million annotations created from that point forward. So it has it now. But what we also do is we do a lot of automatic annotations. So we run around 15 pipelines to do automatic annotation. One of those pipelines, like InterPro to Go, Paul already mentioned that one of the de facto standard for the automatic annotations. Apart from this, though, we run so many other ones and try to combine and enrich that coverage of different genomes to make sure that people who do research in that specific form with that specific genome can also benefit from getting the Go annotations for that. So for those 15 pipelines, we have multiple checks and tests that we also run. So we try to make sure that our annotation the highest quality. So, and that I will show you on a couple of slides, like how we actually work around with annotations, what we do, and how we figure out of it. But I'll not be emphasizing my talk on this. I will emphasize only on results that come from TreeGrafter itself. Right, so the TreeGrafter, Paul had a very nice introduction to it, so I'm not going to talk anything about this at all, and I'm just going to move on to how the TreeGrafter pipeline is implemented in our database. So as I mentioned already, we have something that Protein to Go, our curation tool, right? But we also have two other resources that we support: is a QuickGo, our annotation browser, and Go FTP site that we release all our data. So as a central system to kind of interact with other groups, just for this specific pipeline, we have at least 4 groups involved. We have Ontology Consortium, the UniProt, InterPro, and the PANTHER. So all these 3 groups kind of work together with Go in that chain when we acquire data that we provide to them, and we also provide them feedbacks and reports so they can go and improve their way to generating data. And then we kind of get this new updated data set in improved quality in our site as well. So InterPro, for example, has the InterProScan, which Paul mentioned that you can download and use yourself. But what we do with InterPro as well, that we have a collaboration when we exchange actively data between InterPro and us in order to for Interpro to provide us, let's say, new results. Instead of us running this InterProScans, they have a pipeline that run it for us. And as a feedback, we give them our checks and show them what they can do with the data that they generate to improve their pipelines or do something to enrich the space that they provide. So let's just go to the quick results for TreeGrafter, because this is something that was presented at Go Consortium last time as very, very new. So this data was literally done like week before the Go Consortium. But now it's settled and we have actually already PANTHER 18 data in it. I did not change this slide to PANTHER 18 data because it adds just another 15 million annotation on top of it, but did not change the big picture of it. What you can see here on this slide that from 347 million that data mappings that Interpro give us, we start to go through our pipeline and filters and checks and so on and so forth, and we reduce that set. So we also remove annotations that's more generic, so we don't really need them in our pipeline, right? So we like want to see more specific annotations to our genes, and TreeGrafter is actually very good at it. I will later on show the slide that shows you the specificity of TreeGrafter. It's very similar to Paint, and it's pushing this into that very good manual annotation space actually. So but just look at the filters and all the filtrations. We have still brand new 280 million annotations, from which almost like over 200 million we have not seen before, right? And we have 1.1 billion annotations in total. So this is something that really get excited, like, you know, really get us excited. And I start kind of looking into differences between TreeGrafter annotations and the others, and just by looking at that donut of different biological processes, molecular function, and cellular component, you see that the molecular function, the TreeGrafter really enriched the molecular function. No, sorry, the other way around. Our annotations is enriching molecular function. The TreeGrafter is actually doing a better job for the cellular component and biological process, right? And this kind of leads to kind of thinking about, okay, so what else it does? What else can we compare with? So I talked to curators and they gave me a couple of suggestions. Of course, I did some comparison with other pipelines that we run for automatic annotations, and just, you know, simple few Venn diagrams to show you how the overlaps between TreeGrafter and other Go data pipelines works. And what surprisingly was that the overlap between, let's say, InterPro and TreeGrafter pipeline wasn't that great. So they had pretty unique annotation between them. The same that we have, it looks like it does really good, give us a lot of new annotations, something that we have not seen before. And again, this is like we start looking at these results and we keep going, and we can keep looking at more. So we start looking at some specifics, some annotations like the RIA or Paint itself, how they overlap with each other. The most exciting one was suggested by actually for me to take a look as Valerie Wood from POMBase. She said, Alec, can you take a look at the overlap with the fungi and the TreeGrafter annotation? So you just take all you have and see what happens. So I took all the annotations that TreeGrafter predicted for fungi, and all this stuff that we have predicted for everything else in Go, and we have about 50 million in Go, and TreeGrafter gives an additional 21 million annotation, which is a big enrichment in this specific site, right? And we really like to see feedback from more curators once we get this data public and see what they think about it and how the groups will react to this, to be honest. But we were talking about specificity. So, and I just plot some general distances. So what about specificity for me? I was thinking, okay, how do you determine specificity? So, and I come up with very simple method. Literally, it's just you take a distance, a minimum distance on the graph for your go term to the root node, right? And you just plot your averages. So if you ignore annotations, just take gene ontology as a whole, and that's what I call go graph, right? And then I look at the manual annotations and where the manual annotation sits, where the other pipeline sits, and you can see that TreeGrafter is literally one of the best pipelines that we right now have for specificity. The only thing that beats them is RIA2Go, which is understandable, and Paint, which is really manually created pipelines. But everything else that we have, InterPro, Unirule, which is like comes from specific rule-based pipeline from Uniprot and others, is even machine learning pipelines like Arbor2Go is not performing as well in this respect as a TreeGrafter. So it's very nice and very exciting results. And we would like to kind of get it as fast as possible out, but we also was careful to do more testing to get through more tab. But what I can tell you that this is what's coming. So the annotations available since October in Protein2Go, but this week I'm planning to release them in Index and with QuickGo, so they will be available for everybody to see. And as release progresses next week, we're going to publish all this data for all the species that we have on the Go FTP site. So that's pretty much all presentation for me. And I want to thank everybody who was involved in Goa TBI and Go Consortium. Specific thank to Valerie Wood and Pascal, who helped me a lot to kind of work with the taxonomic constraints on the PANTHER data and so on and so forth. So, but as I said, we are very excited to see it coming next week, and hopefully people will get more feedback to us. Thank you.
Thank you, Alex. Thank you, Paul. So I want to open the floor up to questions. Like lots of applause. Do you have any hands up for questions from the audience? I have a question for you, Alex, about so the increased number of annotations that you find with the TreeGrafter. Is that like more specificity? So like if you sort of looked at the hierarchy and what you're sort of getting is an expansion of more granular annotations, like but if you sort of, you know what I'm saying? It's like, you know, if you're looking at the sort of regular Goa annotations, that they might be at these really high levels, and what you're seeing is really an expansion, or is it like totally new areas of the ontology? Both. Okay.
It both. Right? So there is some of them that more specific than the other automatic annotation pipeline predicts, but there is a lot of them that we basically see first time.
Fiona? You got to unmute.
Sorry, I thought I had. Thank you both for your talk. My question, and I'm not sure who this should be directed to, but it's about TreeGrafter and the taxon constraints, because the taxon constraints were always relied on us accidentally finding we'd done something dumb and getting those rules in place. So with using those phylogenetic trees, does that sort of take out that need? Because with the phylogenetic trees, you're seeing better cases of when not to propagate an annotation?
So yeah, the number of failures, it looks like, Alex, were you know, due to taxon constraints, weren't huge, but there still are some. We actually tried implementing this two different ways during the so we had taxon constraints. Actually, they're built into the software, this Paint software that we use to annotate the trees. But we found that that the curators often wanted to override it. That the taxon constraint was sometimes overly restrictive. And so yeah, so we're actually doing a lot of work in Go now on refining the taxon constraints and trying to make that a little more manageable. But yeah, but there are still cases though where occasionally, we do find a curated model that made an assertion that couldn't have been possible. Say some multicellular process arising, say, in some unicellular ancestor. So there's still cases where there's a little bit of overpropagation. And so the taxon constraints, we use them basically to go back and find mistakes like this and correct it.
Yeah, I think I also can
So,
add
so
to
yeah,
that.
so it's not perfect, and that's the taxon constraints. Actually, they're not always right, but sometimes they give really useful feedback for things that are wrong.
Yeah, I can also add that this almost happened to every pipeline. If you compare, let's say, taxon constraints that I would report for other systems compared to TreeGrafter right now, they will be probably 10, 15 times less. Reason why? Because I explained that we have this loop of exchange of data. We feed it back to the groups, and groups sometimes can exclude certain things and remove. Like the latest, like with Interpro, we worked for this for like 10 years now, and literally the latest taxon constraints for Interpro almost give you nothing, right? But there is still sometimes, and this feedback, because every time we process our pipelines, which is about every 8 weeks, right, to regenerate all these 15 pipelines with all these billions of annotations. Every time we do that, we also check all the taxonomic constraints and send that feedback to the groups. And by default, we delete them all. So we don't want to have any violations that will be released to the public, right? So this is kind of a nice thing about doing this often and give it feedback to the groups and get back from the groups, and it just improve the whole process.
Just a quick follow-up question, Alex. So is the taxon constraints run automatically and integrated with the InterProScan, or is that something you run separately?
This is something I run separately, yes. This is
Okay.
after all the pipelines are run, all the
Yep.
data is checked on the taxonomic constraint.
Thank you.
You're welcome.
Yeah, so that's a really good point, Fiona, and I think that that's one reason to try to get these results directly from UniProt when they're available, because they're going to have that extra level of constraint on the annotations that actually get released.
Any other questions before I jump in and ask more questions? So I'm just looking around at the audience too, and of course with my own particular bias of being a plant person and wondering about, because I know that so much of the paint annotation is focused on like human-centric gene families, and wondering like what the performance is for plant-specific taxa. Because it relies so much upon having the experimental Go annotations, and I know how much we're able to or not able to do for Arabidopsis. And I don't know the extent to which UniProt has, what all the different plants are that you guys are manually curating. But I do wonder, like, are there differences that you see between in like plant genomes versus animal genomes? Did that make any sense?
Yeah. Alex, have you looked at comparing the numbers for different taxa? Like what
No, I
what the
didn't.
coverage looks like for, say, an average plant compared to an average animal?
Yeah, but this is because I never done a release with TreeGrafter yet. So it was
Yeah.
just kind of kept in our database. But this coming this next week, literally early next week, we should get all the numbers more or less for the major model organisms that will go out first. And there's also going to be again, I'm very flexible. If somebody wants, knows this number tomorrow, just let me know what taxon you want to look at, because looking at all of them will be kind of crazy. But few of them I can query manually in the database and provide the numbers.
Yeah. I was actually hoping so I've got someone in my group looking at trying to get these numbers for some representative genomes. I just wasn't able to get statistics in time for today. But I can just tell you anecdotally, I was surprised when reviewing the kinds of, so what I did is got lists of biological processes that are well covered, and a lot of plant development terms came up. Those are really highly annotated in these trees. Photosynthesis is in the list of ones where we have a lot of genes that are so just anecdotally, I was surprised to see, and they're fungal specific processes as well that I was a little surprised to see so well represented. So I'm curious to see what those numbers are too, but I think they're not going to be bad coverage for all taxa, because really these protein families, as I said, so even though the effort has been on annotating families that have at least one human member. That's been our first goal. The truth is, what we do is we don't just go and annotate the clades that have a human gene. We do the whole family. And so you have like some of these massive, say, protein kinase families, and there are members from all over the tree of life, and all of those get equal attention, of course, according to the annotations that are there. So for extrapolating to the plants, we rely really heavily on the manual annotations from TARE. And so this has really been but I will say that again, the coverage is pretty good, especially across eukaryotic genomes. I'd say less so for prokaryotes, just because our major sources of manual Go annotations are E. coli, and then a number of prokaryotic genomes that UniProt does some manual annotation for. So all of those are imported into this, but the diversity among bacteria is much higher. So I'd say for eukaryotes, we're probably doing a pretty good job across the eukaryotes, but don't believe me, believe the numbers that we'll be generating soon.
Are there any other questions from folks in the audience? If there's nothing else related to this, I did want to ask, so for those of you who haven't seen it yet, Alex's group also published this really nice little paper on quantifying the impact of your databases. And do you mind just like really quickly, Alex, sorry to kind of deviate from this, but I think since I have you here, and I think it's something folks might want to take a look at. Alex, could you like quickly just sort of tell us about what that project is?
Yeah, I mean, basically it's something that Alex Bateman originated. He has to provide this for the reports all over again every year. Has a bunch of scripts that he wrote himself in Perl, and he tried to manually do a lot of work every year for this. So, and this project is literally, we just say, okay, well, how about we do something useful for the whole community? Because it's not like just Alex doing this, it's everybody doing the same thing. They need to know what literature mentioned there, resources, what grant agency actually provide grants to those to those groups, and so on and so forth. So how many grants was published and mentioned UniProt, for example, and stuff like that. So, and we basically come up, okay, so we have so many resources at TBI. There's so many groups. We have EuroPMC, we have Campbell, we have which has huge archive of data for the patents and stuff. So we basically say, okay, how about we investigate this a bit more, and using local resources, we'll try to create some sort of program that will allow people to do this. And we publish it on using Google. The Python is called Colab. Is literally like your Python notebook, but online. And anyone can basically go and do the drop down or type certain things that he wants to know, click and get some figures, some results, download them, and then use later on for whatever reporting they want to do for the grant agencies or for so specific you know, I don't know, consortia that they want to do. So take a look at the, I think it's very simple. If you're familiar with Python and know how to program, you can actually even do it yourself. You can have your local versions, modify them, and so forth. We had a lot of interesting questions that we have to kind of go through. Like some people say, well, you guys include all this and age agencies, but we're in South Africa. Can you guys please include all South African agencies too? And we're like, yeah, how are we gonna do that, right? So we have to have a talk with people from Europe PMC asking, guys, can you give us something like this? They said, we don't have anything like this online, but we can give you a file. Okay, give us a file, then we will put it in, and maybe we'll put all of them. And then they give us a file of about, I think it was 40 or 50,000 agencies in total for the whole world. And we're like, we're not going to be able to put this all in. So let's just look at least some most popular one that has most hits. So and that's how we kind of basically try to come up with. It was a little fun project. I did not involve a lot into the programming. My part was mostly like investigating and find those resources, bring it over, which API we're going to use, and so on and so forth. We are very good Python programmer. I don't need to really step on his his work.
No, I actually I had a little bit of fun with it too, like sticking and tear and trying to look at those metrics. So I just thought it was really interesting. I just wanted to make sure that people were aware of that. So last chance for any questions from folks. I just really wanted to thank you both for coming and giving this presentation. I know that, you know, you guys, these are both very heavily utilized resources. And so I think it was really great to share the latest updates, and especially about like all of the TreeGrafter stuff, which, as you know, we're very enthusiastic about at TEAR. So last chance. No more. If there's no more questions, thank you both very much. And
Hey,
if
thank
there's
you so much for the
Thank you.
invitation. It was
Thank
great
you.
to get a chance to share this with everybody, and thanks for the great questions.
Yeah, so everybody go and download your annotations. Okay. All right.